just about.
So I think I do know everybody, but I'm Brandon, and I'm presenting a little bit of an overview of my research today. And I'm in the Aggie lab, my mentor is in the back, over at the Helen Diller Cancer Center. So just to give you a sense of what I hope to accomplish today, I have 3 goals. The first is I just want to give everybody an introduction to glioblastoma, as well as tissue hypoxia, which is a driving force of that tumor's invasive characteristics. Then I want to talk a little bit about some of the background that my lab has done, which got me to my current project, affectionately known as Autophagy 101. And then talk a little bit about the design of the project that I've been working on and some of the initial results that we've received. So pretty much everybody knows about GBM. It's unfortunately one of the most aggressive human cancers. It's a high-grade malignancy of the astrocytes, which are the structural glial components which surround the neurons in the brain. GBM is the most common, as I mentioned, along with anaplastic astrocytoma, occupy about 40% of primary brain tumors. And it's an extremely aggressive tumor. The median survival is about 15 months. And something which attracted me to the disease initially is that this figure really hasn't changed much in the last decade at all, with a pretty dismal 5-year survival of less than 5%. And the therapeutic options remain limited at this point. The standard of care is maximal safe resection, followed by temozolomide, and then X-ray therapy. And most med students are familiar with the board's picture of the ring-enhancing lesion in the brain, and that's GBM. So one of the observations that's been made is that angiogenesis is actually highly upregulated in GBM. So it's thought to be due to the fact that the tumor itself is so hypermetabolic, using all of the nutrients, that creates hypoxia or lack of oxygen, which then triggers numerous signaling pathways, including VEGF, which lead to more vessel development. But the interesting observation which has been made is that even though these tumors are really bloody and have a lot of vessels, these vessels are actually quite abnormal. So they're tortuous and they don't work like you would expect normal vessels would work. And this fact actually has a number of clinical consequences. So the fact that they're leaky creates vasogenic edema, some of the mass effect, which creates the symptomatology in GBM. Also, from a clinical treatment perspective, we have a tough time delivering chemotherapies to these tumors, and also the fact that they're not getting regular blood means that this initial hypoxia becomes even worse because they're not able to get new supplies of oxygen. And that hypoxia would then lead to reduced X-ray therapy sensitivity. So all in all, kind of a complex brew of forces leading to this tumor being so hard to treat. However, a few years ago, there was actually some enthusiasm that there might be a new age in GBM therapy. The idea that you can target this angiogenesis with an anti-VEGF agent known as bevacizumab or Avastin. And this agent was approved based on 2 phase 2 trials because they were so overwhelmingly positive. Just to call your attention to them, historically, patients have a 6-month progression-free survival, so a time from stable disease to the first radiographic evidence that their tumor is going to recur of about 15%. In these trials, it was about 2 to 3 times that, so the FDA decided to approve this medication conditionally. And in patients for whom they are treated with Avastin, the results are often quite dramatic. This is an MRI from one of the initial phase 2 trials. This is a patient who received 4 doses of cycles of Avastin. You can see it looks like the tumor almost melted away. And this radiographic improvement is often coupled with actual clinical improvement. These patients do cognitively better, they feel better, they look better. So people were quite excited about these results. But unfortunately, that enthusiasm was tempered by the fact that 40 to 60% of those patients actually developed resistance while they were on therapy. So they progressed while being treated with Avastin, suggesting that their acquired therapeutic resistance would actually be quite a significant problem for this agent. And one of the interesting observations that people in my lab made was that if you look at the cohort of patients who were treated with Avastin, they actually end up doing poorer than patients who weren't treated with Avastin at all. So just to reiterate that, they do better in the short term, but then they do poorer in the longer term. And there are a number of ways to categorize why they do poorer. They have poorer overall survival, and there's a Kaplan-Meier curve where you can see that patients who were treated with Avastin are actually quite separated from surgical patients who hadn't received Avastin. They have worse progression-free survival as well, similar Kaplan-Meier curve. And not surprisingly, because you're interrupting the vasculature, they have worse perioperative morbidity if they undergo a second resection of that tumor. So my lab is actually quite interested in some of the mechanisms of Avastin and other anti-angiogenic agent resistance. So people think of resistance in two ways. You can have a tumor that never responds to an agent, or what I'm talking about is a tumor that initially responds, so it gets smaller, but then it comes back. And what we've observed is that when it comes back, it's more invasive, it's more malignant, and those patients tend to have poorer clinical prognosis. So I think it begs the question of, we clearly haven't figured out the ideal way of using these therapies yet. Is there some sort of synergistic strategy, or is there some other way that we can use this agent to actually make it more effective for GBM?
When you say, we're talking about the Avastin, were those given as neoadjuvant, like prior to resections, or?
So they would have an initial resection, and then they would go beyond the Avastin. There have been a number of trials. It's been tried in a few different ways. So there are a number of different mechanisms that have been proposed for why cancers become resistant. One is potentially neovascularization. If VEGF is being blocked, maybe other factors are being upregulated that can kind of trigger the development of new vessels. Perhaps they're recruiting bone marrow markers which are able to grow new vessels. Maybe there's increased pericyte coverage of the vessels, so they essentially form a shield that the Avastin can't work on those tumor vessels. But the one that's most interesting to me is perhaps these tumors are upregulating genes which help them to become more invasive. So and I think this makes sense from an evolutionary perspective. If you treat a tumor or you treat any cell, you make it a harmful environment for them. In this case, we're shutting off the blood supply, making it hypoxic. It would make sense that this environment is now no longer hospitable to cellular growth. So perhaps evolutionarily, these tumors have developed a way to escape that local environment and move to places where the nutrients might be more plentiful for them. So how did we get to the project that I'm currently working on? The lab actually took a look at patients who grew resistant to Avastin. And these were patients who had tissue prior to being on Avastin and after being on Avastin. They're all surgical patients. And we took a look at their radiographic images for the first go and saw that all patients initially, they actually had the classic radiographic image of GBM, so these T1 bright enhancing lesions. And then if you looked after the tumors recurred, a handful of patients had that same pattern. We called those the enhancing cohort. But there was also, interestingly, a cohort of patients that had a very different looking radiographic pattern after being treated with Avastin. These were patients who were T1 dark, where they weren't enhancing, but they were enhancing on the FLAIR sequence of their brain MRI. We call those the non-enhancing gliomas. Then the lab did a number of studies looking at those tissue specimens of the enhancing versus the non-enhancing cohort to see if there were any differences. And what they found was that the non-enhancing group actually had lower vessel density based on lower von Willebrand factor, increased hypoxia as evidenced by HIF1 and also CA9, whereas the enhancing cohort, basically there was no change in rate post Avastin. When they looked at cellular proliferation, which is a big important thing to look at in cancer, now the enhancing group seemed to be much more proliferative after being treated with Avastin, whereas the non-enhancing, there was no change. Then they looked at cellular invasion, or the ability to go through the extracellular membrane to adjacent tissue, and now they saw that the non-enhancing group were much more invasive than the enhancing counterparts. And finally, they did a microarray analysis where they looked at various genes which were up or down regulated in the pre and the post-Avastin treated tumors, and actually found that in the enhancing cohort, there was upregulation of genes which led to increased tumor proliferation and neovascularization, whereas the non-enhancing group, they became much more invasive. So we made the observation that these non-enhancing groups, they might be more hypoxic, they're more invasive, and we also saw a genetic signature that they might be more invasive. And from those observations, the lab made a hypothesis that this hypoxia is actually quite important for driving some of this resistance. There have been a number of reports over the last decade that have shown that hypoxia and other cellular stressors can actually upregulate a cellular process known as autophagy, which I'll talk a lot more about in a few minutes. But numerous stressors have been shown to activate autophagy, including many of the agents that we use to treat cancer. So what autophagy is, a lot of people are familiar with apoptosis, which is a cell death pathway which is triggered when the cell becomes so mutated or so damaged that the thought is that it can no longer continue, living. Autophagy is interesting because it sequesters off only the parts of the cell which are damaged, but the remainder of the cell is actually able to persist. So instead of a cell death pathway, it's actually a cellular preservation pathway, eliminating some of the harmful sort of pieces of that cell. So we all have autophagy going on right now at basal levels. You can think of it almost as a tumor suppressor, because if there's a mutation that I get that damages one of my organelles, my cells are able to recognize that, sequester it, get rid of it, but the rest of my tissue is able to continue unaffected. However, the thought is that once a tumor exists, it's able to hijack autophagy in the same way, because we're using a lot of these agents to create damage in these cells. The hope is that this damage would lead to cellular death, but perhaps the tumors are using autophagy to basically continue to live in the face of the cellular stressors that we're generating with the therapy. And I think of autophagy, and many people think of autophagy as an adaptive response to a stressful cellular environment. So just to talk a little bit about what autophagy is, this is a really hot topic in medicine now. I hadn't heard about it until working in this lab, but New England Journal just published a review on it. So this is an area with pretty broad applicability, not just in cancer biology, but in infectious disease and cardiology. A lot of people are studying it. And just to walk you through kind of the overview, there are 4 different steps. But what happens is there's the formation of vesicular membranes around damaged organelles, which form autophagosomes, eventually fuse with lysosomes, which lead to the destruction of the intracellular contents, and then the breakdown of those vesicles. As I mentioned, there are 4 different steps. I don't want to get into the nitty-gritty, but suffice it to say that there are different genes which have been associated with each of those steps. And a couple that I'll just highlight are ATG7, or autophagy ATG7, and LC3. Those are genes that I'm going to talk about a little bit later in the presentation. So about a year and a half ago, our lab started studying the connection between hypoxia and autophagy, and actually found quite nicely that this hypoxia, as predicted, is leading to an upregulation of autophagy. And trust me that BNIP3 is one of the genes that actually is a marker of increased autophagy. And these are cells here, this is a Western blot, so showing protein levels. These are various glioblastoma cell lines grown in 20% or normal oxygen versus 1% or hypoxic conditions. And you can see in all of the hypoxic conditions, there's a nice upregulation of autophagy. They also did some in vivo studies in mice where they looked at treating with Avastin or bevacizumab and Avastin plus chloroquine, which is actually an autophagy inhibitor, and saw a separation between those 2 curves, which gave them the sense that perhaps autophagy is actually contributing to some of that resistance that is developing to Avastin. Yeah?
Maybe you already said this and I missed it, but is this breakdown of the group that had a more invasive picture after treatment and the group that had the same classic hearing GBM, is that associated with their clinical outcome? Like you said, the VEGF treated group had worse clinical outcomes.
Yeah, so
as like a mean, but would there be like a bimodal?
I'm actually not sure. I would assume though that the patients who had that non-enhancing pattern would do worse than the enhancing ones. But what have you seen in your
It's not statistically significant,
Okay.
but slightly worse, but not
This was just another demonstration of it, so 2 different cell lines. So that paper, it posed some interesting thoughts that autophagy might actually be driving some of the resistance in bevacizumab-treated glioblastoma, but it also asked a number of questions. So at this point, we knew that GBM that was treated with some of these agents is hypoxic. We know that this hypoxia triggers autophagy. We know that disrupting some of that autophagy seems to overcome some of that resistance. And separately, I showed you a few slides ago that GBM that is resistant to these anti-angiogenesis agents shows the capacity for more invasion. So we tied this all together and said, perhaps autophagy is not only allowing these GBM cells to survive, but maybe it's endowing them with an even more malignant phenotype, enabling them to become more invasive and more migratory. So that's really the crux of what I'm studying this year. So my initial aims for the CTR fellowship, the first was to basically just answer that question. Is autophagy driving increased malignant or invasive or migratory behavior? The second question was more of a biochemical inquiry. You know, if this is in fact happening, what are some of the signaling pathways which are involved? And there's been a lot of research that shows that the mTOR pathway is contributing to this autophagy. So that was our second question. Can we figure out what exactly is going on? And then the third aim, which I was excited about, was can we use pharmacologic inhibitors of autophagy to either be synergistic with existing cancer agents or even alone, would they be helpful in the treatment of cancer? But as we thought more about the paper which was written by the former member of my lab, I think it raised a number of questions. You know, the first is autophagy actually driving this invasion and fundamentally driving a change in tumor biology? I think this is a bigger question than just bevacizumab-treated Avastin. Autophagy is really changing the cellular machinery and making these cancer cells that much more invasive. That's a bigger question, and I think it has broader applicability than just GBM. It's potentially all cancers that grow resistant to therapy might think about having to overcome autophagy. I think the second question that we asked was, so who cares? If the tumor has more autophagy, does that even matter for patients? You know, we had hints that maybe they're going to be poorer, but patients with high levels of autophagy, do they have a poorer clinical prognosis? So we wanted to figure out patients that do worse, is there more levels of autophagy in those tumors? And then finally, if autophagy is upregulated, what's the best way to inhibit it? So that initial mouse trial that I showed you used chloroquine, mainly because that's what's been used in the literature and that was readily available, but is that the best way to do it? There are a variety of other autophagy inhibitors in development. Can we think about optimizing that? So those are the sort of three questions that I've set out to answer this year. And kind of thinking about multiple pieces of this project, the first to answer the question, is autophagy even related to invasion? My goal has been to develop cell lines that either upregulate or downregulate autophagy, and then to perform assays to see if those cells are more or less invasive than controls, more or less migratory than controls. And we're also going to develop some fluorescent cells that enable us to track various autophagy markers, and we can say if the cells that are more fluorescent, i.e. more have higher levels of autophagy, are more invasive, that gives us more evidence that we're going to be able to make that correlation. The second piece of the puzzle, as I mentioned, is really interested in what is the prognostic implication of autophagy. So the way we thought about addressing this is our lab actually has access to a very rich tissue bank of surgical specimens of patients who were naive to bevacizumab or treated with bevacizumab. So we thought it would be interesting to stain a variety of autophagy-related markers, quantify them and see if there are high or low levels, and then see if we'll be able to correlate that with how they did, both in terms of overall survival and progression-free survival. I think it's interesting from a prognostic standpoint. You know, if you can say that there's a level of a certain protein which is really high in all the patients who do really poorly, that's interesting. You can give families guidance based on that. But it also informs potentially future drug discovery efforts to say, if this is something that's really important for resistance, can we target it? Can we block it? The third piece is the small molecule study, as I mentioned, and I think the big question there is, what is the best way to inhibit autophagy? So can we optimize the chloroquine dose? Quinacrine, which is another anti-malarial, has also been shown to be an autophagy inhibitor, and there's some anecdotal evidence that that one might be better, but it hasn't really been tested yet, so that's something that we hope to do. And then finally, I think bringing this all together, this is the basic science effort. I'm probably most excited about how we can use this translationally, and we're hoping to develop a mouse study pretty soon where we're going to combine either bevacizumab or another anti-angiogenesis agent plus whatever optimized small molecule we were able to figure out from this part of the work. And then the ultimate goal, treating mice is nice, as Dr. Harris said, but is to develop a protocol for a phase 1 human clinical trial where we would combine Avastin plus one of these agents. And that was something that I started in the DCR class earlier this year, so it'll be nice if that comes to fruition. So just talking a little bit about each of these sort of areas of the project. As I mentioned, my goal has been to develop cell lines that are modulated in their ability to have the levels of autophagy. So to inhibit them, we adopted the approach of using genetic knockdown, so SH small hairpin RNAs, which block the expression of various proteins. To activate, we can either culture these cells under hypoxia or use a small molecule trifluoperazine. And then once we have these cells, which have varying levels of autophagy, we have identified a number of assays that we can test the various components of cellular migration. So first, the cells need to adhese, and there we will plate cells on synthetic extracellular membrane and use crystal violet to see if the cells with knockdown autophagy are less adhesive. Similar with motility, we've identified a scratch assay where you plate cells and you draw a scratch down the center, and you see if the cells are able to migrate towards the center. And then for invasion, we've identified a number of assays where we will see if these cells are able to pass through synthetic extracellular membranes, which is thought to be a model of metastasis. So just to give you a sense of where we're at, we have developed a number of knockdowns for various stages of autophagy. We developed 6 different cell lines with shRNAs, one at the vesicle elongation stage of autophagy, and that's the ATG12 gene. We have 2 different hairpins for that one. For the vesicle maturation stage, which comes a little bit later in autophagy, we have 2 different genes that we've been able to knock down. And then we also have a non-targeting control that we're able to compare each of these cell lines against. And we've developed actually a ton of viral stock of each of these shRNAs. In the literature, it's been shown that autophagy is so important to cells in general, but even more importantly to these tumors, that they actually very quickly develop the ability to evade your knockdown. So it's important to remake these knockdowns quite soonly before you're going to actually run these functional assays. So we will do that. And just to confirm the knockdowns, for ATG3, you can see this is a Western blot compared to a non-targeting control. The hairpin, it looks like it's a pretty good knockdown. For ATG7, we tried 2 different hairpins, and it looks like this number 84 is giving us a better result. And then HEG12 has been sort of the bad sibling of the group. I've had a harder time optimizing some of the Western results, Western conditions for that, but at least some of our pilot studies seem to suggest that it is knockdown. And they're behaving very similarly to the other cells, so I'm optimistic. This is kind of a messy blot for the bio folks in the audience. So the second part, which is the staining piece, just to give a sense of what has been done so far, we conducted a literature search of all possible genes which can be related to autophagy, got it down to an initial list of about 10 genes that we wanted to stain in a pilot fashion, and then I was able to stain some frozen specimens that we got from the tissue bank, and also some GBM cell lines. And from those initial results, as well as a couple other factors, which I'll explain, we prioritized down to a list of 5 for initial study that we're going to go forward with. So next steps there is we're slated to receive 10 to 20 cases of naive GBM, so these are patients who haven't been treated with any agents yet. We thought it would be important to check at the basal level, how important autophagy is. Then we will stain them against these 5 genes that I will, 5 proteins that I will mention. And it will be important to optimize the quantification approach, because I think anytime you do immunohistochemistry, you stain and you make very pretty pictures, but it's often hard to make something out of that. So I think we're going to have to think about how we quantify the results so that we can actually correlate that to some of the survival data that we're also hoping to do with. And then finally, if we develop promising candidates from this small study, the hope is that we can expand it up to about 100 cases to establish statistical significance. So these are the 5 genes that we've selected to go forward with, and the NIP3, which I showed you in a Western blot earlier, ATG 6, 12, 7, and then LC3, which was a key protein for autophagy. And these were selected for a number of reasons. 1, they worked really well in the pilot study, so it was reassuring. 2, we were conscious of trying to include all different stages of autophagy, everything from initiation of the process itself all the way to maturation of the vesicle. And then finally, there were some correlations to some of the basic science work we were doing. Some of these other candidates are actually being studied in other cancers in the literature, so we thought it would be interesting to study in GBM as well. And as I mentioned, if we find that some of these are upregulated, that may inform future directions of the lab in terms of drug discovery efforts. So this is what the pictures will look like. This is one of the pilot studies in some of the frozen tumors that I did, and these are 3 fields from the same tumor slice. And this is BNIP3 and ATG7, 2 of the genes. I don't know if it's obvious from this picture, it's not a great image, but there's a lot of heterogeneity depending on where you look in the tumor, and this is a challenge, I think, with immunohistochemistry and with this study as well. So we recognize that this may not yield a very consistent result, so what we've decided to do is actually we're going to get tumor chunks in addition to the staining, and we're going to run Western blots for these markers as well, so hopefully we can correlate the Western blot result with the staining result. Yeah.
Sorry, I mentioned this as well, but are these genes coming from microarray studies that you mentioned earlier? So were these like differentially expressed in microarrays and now you're...
So they weren't explicitly tied to microarray, it was more of a literature search of what is involved in autophagy. And this is a picture of what it would look like in the immunohistochemistry and cell lines, similar picture. Here we just stain for phalloidin, which is an actin component, so it just enables us to make sure that our staining is actually within cells. And this is not the best quality, but you can see these sort of like punctate looking stains, and that's the classic appearance of LC3 in autophagy, so that gave us a good sense that what we were doing was actually real and not just a background. So finally, as I mentioned, the part I'm most excited about is using small molecules and trying to optimize what is the best way to inhibit autophagy. In those initial studies that my lab conducted, they used chloroquine, as I mentioned, because it was convenient, and there have been actually now about 20 clinical trials in a variety of cancers that are testing chloroquine or hydroxychloroquine. But just because everybody's doing it doesn't necessarily mean it's the right way or the best way to do it. So through discussions that my PI has had with other mentors, there's been a discussion that maybe quinacrine might be a better way, it might be a more potent autophagy inhibitor. So that's a question that we're trying to tackle. And maybe we have to go outside of the antimalarials. Maybe we have to identify other things too to get it to really work. So to explain a little bit how this will work, I just want to go through quickly autophagy one more time. And as I mentioned, you form these autolysosomes, which lead to the destruction of cargo or damaged organelles. And there's a particular protein that's pretty important to this process known as LC3. And that was one of the markers that we're staining for as well. LC3 exists in a number of different forms in the cell. Pro-LC3 in the cytoplasm is then converted to LC3-1, which is also cytoplasmic, and that's the start of autophagy. And then as the process becomes more mature, LC3-1 is converted to its mature form LC3-2, which is then membrane bound. So LC3-2, the reason why people stain for it is because it's found in the vesicles of autophagy. The thought is that if you allow autophagy to go all the way through to completion, eventually this vesicle is going to break down everything internal to it, but also the vesicle itself breaks down. So you would expect this LC3-2 levels would degrade if autophagy was allowed to continue to completion. We're testing the role of chloroquine and quinacrine. It's believed to inhibit autophagy at this stage in the process, so preventing the lysosome from fusing with the autophagosome. And if you allow this to block this stage, you never get to the autolysosome step. In fact, if this was working, you would see LC3-2 accumulation. So we used LC3-2 accumulation as a marker of how potently we are inhibiting autophagy. And just to kind of tease with some initial results, we looked at optimal sort of dose ranges and as well as LD50s in 2 different GBM cell lines, and then grew the cells with varying doses of chloroquine and quinacrine. And much to my delight, you can see that for LC3-1 and LC3-2, as we go to higher levels of chloroquine, you see more of LC3-2 compared to LC3-1. So we are accumulating the marker that you would expect if we are inhibiting autophagy. The quinacrine results didn't look as great in this cell line. It seemed pretty uniform across the board. Though I will say that if you look at from a kind of a pure pharmacologic potency argument, at 10 micromolar of quinacrine, you are seeing more LC3-2 than you are compared to chloroquine. So maybe it is more potent. We got we were able to repeat similar results in another cell line of GBM. And in this one, actually, the quinacrine did actually look better as you went to higher doses. So in the next WIP, hopefully I'll be able to talk a little bit about some of the results of those various aspects of the project. But we're excited so far. We think